You're listening to the Veterinary Innovation Podcast.
You're listening to the Veterinary Innovation Podcast. My name is Sean Wilkie, and along with my awesome co host, we interview the innovators in this space every week. Ivan, please kick us off.
Hi, I'm Ivan Zach. I'm excited to introduce a guest today, Dr. Christian Leutenegger. He is coming from Antech Diagnostics, where he holds the position of Vice President of R&D. He graduated from the School of Veterinary Medicine at University of Zurich, Switzerland in 1992, where he completed a doctoral thesis on developing vaccines for FIV. Later, he earned a PhD by testing the first DNA vaccines in veterinary medicine. Dr. Leutenegger developed a strong interest in molecular immunology and virology, leading to the early development of protocols of DNA quantification using real time PCR. In 1999, he founded the Real Time PCR Research and Diagnostics Core Facility at the School of Veterinary Medicine at UC Davis and expanded its services until 2006. After 13 years at IDEX Laboratories, where he introduced standardized molecular diagnostic to the veterinary industry, he joined Antech Diagnostics to expand the molecular testing portfolio. Dr. Leutenegger has published over 200 peer reviewed papers and book chapters and has a broad network of collaborators. I bet you do. That's a lot of accomplishment. Welcome to the show.
Thank you. Thank you, Zach. Thank you, Sean.
So, Christian, the extensive molecular diagnostics experience that you have will really help me to understand that as a simple veterinarian on the front lines. Can you just tell us simple veterinarians what have you worked on that is introduced right now and what are you focused on a day-to-day basis at Antech?
Yes, let me start with the latter question. What I'm focused on primarily is to develop new and innovative reference lab diagnostic tests. So, of course, we differentiate between the reference lab type of testing, the in-house diagnostics on the equipment side, and then the rapid assay tests for certain biomarkers or infectious agents. Those tests you can run in the clinic. So I'm primarily focused on the reference lab side, both molecular and serodiagnostics.
Very interesting stuff. And the first thing that came to my mind, Christian, when we were chatting before the episode was you're Swiss, you live in the US. That's an incredible culture change. I lived in Zurich for a little while and in Basel and understand a bunch of the nuances between the two cultures. One of the questions that I wanted to ask you, you're in an innovation role. What are the cultural differences that have been very apparent for you as you've kind of crossed borders from Switzerland to the US? Kind of seems like you got stuck over here. And how has that helped you in your career as a true innovator?
What really helped me coming to UC Davis is the incredible way of collaboration, which became so evident at the School of Veterinary Medicine. When you look at the human side, the human research, the veterinary research, and then, of course, UC Davis being an Aggie, so there is that plant-based or deep research available too. That amazing collaboration really influenced me greatly in seeing across the borders, literally across the border from Switzerland into the US, but also being able to bridge over divides, which normally people see as difficult to overcome. And molecular diagnostics in particular was a very good example, which allowed me to see beyond veterinary medicine, to look into what's going on the human side and to be integrative even with some of the plant applications. So it's really become a foundational aspect, I believe, in my career, but also how I approach the aspects and the projects which I need to find solutions for, which it now, of course, primarily focused on veterinary diagnostics.
So when you are we always talk about the vet medicine being behind the human medicine, but sometimes you find, especially these days and in technology, what I find, or in research, where it goes the other way around. So essentially, some of the early research is done on animals, and then it sometimes is brought first to veterinary medicine. What did you find from your experience crossed over one way or the other? And if you subjectively said, how far is veterinary medicine behind the human medicine? How many years?
I couldn't say that as a general statement. As you correctly outlined, some areas, some topics we are far ahead on the veterinary side, or veterinary diagnostics included, or including, of course, that goes into therapeutics too, right? One aspect on the diagnostic side is that we have fewer levels of regulation to work through. So actually, to bring a diagnostic test into a reference lab is relatively straightforward. And that is a major hallmark of the veterinary diagnostic market, or which should really propel us ahead. But then again, it really depends on the area of application. Or we are behind in certain arenas, probably by a decade, but we are ahead in certain diagnostic applications, probably by about the same amount. And that some of these applications include molecular diagnostics testing.
Excellent. Well, what are the exciting things that you're working on right now?
What we have long been working on is what we call panel testing. So you're not just testing for one analyte, you're testing for an entire series of analytes, of targets. In particular, in molecular diagnostics, very early on, we focused on what is called syndromic panel testing. As a clinician, both of you, of course, are very versed in the clinical entities. A patient is being presented to you normally with a leading clinical sign, like respiratory distress or intestinal upset or scratching, so dermatitis. That's how we approach syndromic panel testing. The classical examples here are the respiratory and the diarrhea panels, where we test for more than just the usual suspects. In respiratory, of course, not everything is distemper. Over the last 10 to 15 years, we made very foundational discoveries of new infectious agents which have become clinically important entities. One of them, for example, is pneumovirus, which causes pretty severe outbreaks on the shelter side. And mentioning shelters and larger aggregation of, in particular, dogs, right, in a very small footprint. Of course, that one additional infectious agent, which now has been very well recognized in dogs and in cats, is Streptococcus epidemicus, which can cause peracute deaths in dogs within 24 to 48 hours. So rapid diagnostic testing, fast turnaround time, or even better, if you can do the testing on site, those have become very clear mantras and hallmarks of diagnostic testing. But having said that, diagnostic testing needs to have a certain affordability. Otherwise, it precludes itself to be used widely in a clinical setting. So we are really seeing a combination of things here. It's not only the research and the discovery which brings us a better understanding of what causes these syndromes from an infectious disease perspective. It's also affordability and turnaround time. Those three aspects are absolutely critical if you want to bring anything with commercial success into the diagnostic arena.
Yeah, that makes a ton of sense. So, in the field of molecular diagnostics, which you're a world expert in, what do you think? What does the next 5 years look like, Christian? Like, what major changes do you think we will see?
Let me reflect on the past 5 years and then we can go over the next 5 years. And what I see major developments will be at the diagnostic front. What we've been able to achieve at Antech, in particular with the intestinal parasite panel, key screen, is we were finally able to progress from simple detection of an infectious agent with key screen, these are intestinal parasites, to actually creating answers which influence the outcome of the patient. So a better world for pets means that you get therapeutically actionable information with your first diagnostic test, not with your second or third, and not 72 hours later, but at fastest possible turnaround time to a price which is affordable and with the therapeutically actionable information, which is we were able to include drug resistance testing into KeyScreen, which now can tell you what type of anthelmintics not to touch in certain parasites, and actually go and use combination therapies which use a combination of anthelmintics from different classes. That's how you maximize the effectiveness of your therapy. And again, most importantly, is you get that information with your first test you submit for KeyScreen. It's built into the test, and you always will get a result whether that particular parasite is susceptible to that drug class or whether it has become resistant.
So when we talk about molecular diagnostics and PCR, from what I remember from school, which was very long time ago, there's a lot of sample handling, and especially in PCR labs, with the amount of false positives, I guess, with the sort of DNA omnipresent. And if you're developing something like that in clinics, practically, how do you achieve sort of the lab environment sterility to run the tests? And is that an issue at all?
With the modern reagents in molecular diagnostics, and of course here we are collaborating again with our major industry partners outside of the veterinary field. So we have a long-standing collaboration with one of the three major human diagnostic molecular companies, Roche Molecular Diagnostics. That's where all the extremely stable reagents are coming from. And we have safety systems built into those reagents, which preclude the occurrence of what is normally, that ever feared term is contamination, particularly with the amplification products. We have now safety systems built into the reagents, which allow us to process KeyScreen in a very simple laboratory footprint. It doesn't require expensive laboratory settings anymore. We have a simple separation between the two steps in molecular diagnostics. And we made this a very, very foolproof application. The reason why I can tell you that is the volume we see clinicians submit samples for KeyScreen testing is so high. If there would be a problem, we would have seen it by now. KeyScreen got launched 2.5 years ago in March of 2022. We are nearing 3 million submissions before we end the third year in the market. So it's become a massive volume-based test, which explains its commercial success. I want to just re-emphasize we work in a very safe environment with pretty much absence of false positives due to technical artifacts.
I've got a question. So I know that the diagnostic business is growing and is key to everything that Antech does. But you have a lot of developing markets around the world where the diagnostic field is not as widely used because of interpretation problems from the veterinarian perspective. What impact do you think this crazy new world of generative AI has? How does its ability to affect the interpretation impact Antech's business in the future?
When we look at the higher intelligence type of algorithms, what they allow us to recognize are connections which are difficult to appreciate with the human mind. These systems can take 4, 5, 10 different parameters into account and make a prediction for a certain disease process. Now, when we look into the larger categories of issues we are running in, not just in the industrialized world, but primarily also in environments where diagnostic testing and medicine is not as directly accessible. The big topics are the One Health initiatives to recognize what's the interconnectivity between animals and humans, people, and the environment when it comes to infectious disease. So the One Health topic definitely is a driver in this arena. And of course, the other driver is the antimicrobial resistance stewardship. Antimicrobial stewardship is a highly complex aspect which can be answered with molecular diagnostics very efficiently. Most of the resistance aspects can be recognized with molecular testing, but When we start talking about these issues with higher complexity, that's where really the augmented or artificial intelligence is coming in.
You touched on the antimicrobial resistance, and I'm just interested. I recently received licenses in two more provinces just with the nature of what we do by building clinics. And each province in Canada had you need to pass the test on antimicrobial resistance. And then when you're on the floor in the clinics, it looks like they make it important to, that's a test that you pass actually to get the license in particular geography. But when you're on the ground doing day-to-day kind of, veterinary urgent care grind, I don't see or feel the significant impact and or the importance of it recognized neither by population or by the clinicians. How big of a problem is it? And is it as argumentative as a global warming that some people don't believe in it? Some people believe in it. Is it something to believe in, or there's fact and we're in danger of at some point arriving to the point where we can't use simple medications that we used to combat simple bacteria?
I think the latter is the case, very clearly. If you look into human or bacteriology, antibiotic resistance has become a very big problem in form of what is normally called hospitalism. Or this has left the hospitals and now really entered the sink in your household, where you find now multi-resistant strains of different types of bacteria. This is a highly under-recognized aspect, and it is important to be addressed from a diagnostic perspective. What do I really mean from a diagnostic perspective? When you have a patient on your table and you need to make a treatment decision, if you get that susceptibility result back 72 hours later, does it help you to make a treatment decision in clinic? It does not really help you, right? You may have to invite the patient back in or prescribe a more specific drug in order to follow up on your diagnostic test result. Let's compare this with an in-house diagnostics test, which could give you susceptibility or resistance data in less than 30 minutes while the patient is still in your clinic. You could forego the broad spectrum drugs and go directly to highly specific drugs in order to address the etiology of the clinical presentation. And that's the bridge we have to be able to overcome when we look forward into the next 5 years. That's the innovation we have to bring into veterinary diagnostics in order to make a difference how you, the clinician, can address a clinical case and how we bring truly a better world for pets. That means we need to be able to influence outcome when you see the patient within that stretch of time, 30 to 60 minutes. While you do the initial exam, you should have all the actionable and important diagnostic information to your disposal.
It's fascinating work that you're working on, and we've run out of time, like every other episode. We've got a couple of questions to wrap up, and thanks for joining us. It was really interesting to hear you speak about something that you're so passionate about. First question is a book, a YouTube, a video, something that you found inspiring that you'd like to share with our audience.
Yes, so going back to your first question, how was I able to actually make that cultural or walk that cultural bridge between Switzerland and the United States of America? I believe very deeply that collaboration, which we need, and the open mind, which is required in order to achieve that, we need to have what is normally referred to as emotional intelligence. And so that's the book I have recommended to my children so many times that they don't want to hear it anymore. But as a matter of fact, just the other day, my middle son took the book with him. I think it landed somewhere in a highly diverse environment, even like at Antech and Science Diagnostics, where we have so many different entities working for the same goal, but coming from different angles. That collaboration and the foundational aspect of emotional intelligence is absolutely important and crucial in order to have success in a system like this. So that's the book I would like to recommend to the listeners and the viewers. It gives great insight to overcome some of the divides we sometimes not even know exist, but we behave around these divides. That's my recommendation to overcome some of these divides and see onto the other side of things.
Very interesting.
And what was the book name? The author?
Emotional Intelligence 2.0.
Correct. Emotional Intelligence 2.0. Yeah.
It is by Travis Bradbury. Great recommendation. I'm actually just added it to my Audible to listen to next. So thank you for that recommendation. And the second question that we always ask: is there another innovator in the industry that you would recommend to be a guest on this show?
So it's very difficult to actually select because the very highly dense team of highly educated specialists at Antech and Science Diagnostics almost makes this a difficult pick. But since you brought up AI, augmented intelligence or artificial intelligence, and how important that development is for us to understand higher complex connections within the medical field, there is definitely one person I would like to recommend, Mark Parkinson, as the next interviewee. He has an amazing insight into many different aspects where AI will be foundationally change the way how we do diagnostics, in particular in the imaging field. He has been with Mars for more than 25 years. He understands the environment here better than anybody else. And the progress he brings into diagnostics is really amazing to observe right now. Again, looking forward over the next 5 to 10 years, we will see some very foundational changes in how we look at imaging and how we read X-rays or digital pictures.
Thank you so much for listening to the Veterinary Innovation Podcast. If you want to hear about our new episodes, please follow us on any social media channel. Also, you can check out our website at veterinaryinnovationpodcast.com. See you next week.